again, to repeat, protein interactions for us in this context are when two proteins actually contact each other. So this glycoprotein in green here, 120, so-called GP120, is in contact with antibodies. So the green one is in contact with the gray, yellow, and pinkish here. And it is also in contact with the CD4, with the blue. While, although this whole thing forms one complex in the sense that at some given time, the whole thing is closed in space. In that sense, there is sort of a proximity of CD4 and antibody 1. They are not actually touching. So for us, protein interactions are only touching. Most people, when they refer to protein interaction, mean associations. Associations is A activates B, activates C, activates D. So pairwise they are in contact. But they are neither temporal contact. So this activation here of D in the end may come long after A activated B. Nor are they in physical contact. So the typical technology that people use, or one of the major techniques for getting high throughput data at interactions, is that you essentially put a bait onto a particular protein X. So you want to see whether X and Y interact, the proteins X and Y interact. And you put a bait, something that will fluorescent a transcription factor binding domain, an activator domain on this one here, and you will see a signal whenever these two come together. So when BD and AD come close to each other, and they will only come close to each other when X and Y interact. And then you see some light emitting. And this is the way people sort of test this on a large scale. Large scale means thousands of proteins at the same time. And then they are sort of filling out these signals. One way of doing it is homology-based inference. So this is the old story that we had, but now it becomes a little bit more complicated. So to just repeat what I mean by the old story. So you have two proteins. Typically, when we are talking about homology-based inference, then we have two proteins, Q, that's the query, and a protein for which experimentally you have some idea about its features. So for instance, you know its localization, you know its interaction patterns. Let's call it E because there's experimental data for that protein. And then typically when there is a sequence similarity between Q and E above some threshold theta, then we infer that Q and E have the same feature. And that's homology-based inference. In this story here, this gets a little bit more tricky. Because in this particular case, we are talking about two proteins, A and B, and we're talking about two proteins that are similar to those, A prime and B prime. So now here is one threshold, theta. In this particular case, you have two thresholds, right? You have the threshold between A prime and A, and you have the threshold between B prime and B. Now, there's a variety in which you can now plot it. You can sort of think about the average, you can think about the maximum or the minimum. What I'm going to show you on the next slide essentially is the minimal, the threshold of the minimal of these two pairs. And I'm going to show you some examples how we can infer, we can exactly use this. So we observe, we know that A and B prime bind experimentally, and we ask, can we then infer that A and B bind? And the answer to that question is shown here, depending on the level of sequence similarity. So depending on this threshold, where the threshold now is the minimal of these two. So you may remember that HSP threshold is this curve here, where at 100 residues, you have sort of 33 or something like that. That's the point. Now, this is essentially the distance from this curve high. So plus 5, plus 10, blah, blah, blah. That's the distance from this curve. So this essentially corresponds to something like 100% sequence identity. And you already see that this axis doesn't show 100%. Nothing near that. It's just a few percentage points. And that ultimately has to do with the problem of the consistency of the data. You do see high error bars, because at the time we did this, is a little bit outdated. We did not have enough data. Today we could do a better job on this. But then it's not interesting enough to repeat things like that. What you do sort of see is that there is some drop. The lower the sequence similarity between these, oh, I have the pair here. So if they are more distant from each other, more distant is here, then the number is lower than when they are more similar to each other. Okay, that much is clear. Now, this gets us to this, what I'm looking at here are proteins in warm. So these two here, for instance, are in warm, and these two are not in warm. Okay, that's the standard idea. Behind this standard idea, there is this concept of homology. The concept of homology, essentially, you have similar parts of a body in different animals, and they're homologous to each other. They may have different functions, such as in the whale here, but they're homologous. And then when you look at genome evolution of proteins, so essentially when you split from species A to species B, and these are sort of supposed to be 3 different proteins, you essentially at the moment of the split, essentially B1 and B2 have the same sort of copies. Right? There's no big difference. They have the same set of genes. And then maybe over time things change. So what I show here is a duplication event. So you take this gene and you duplicate it, not fully, so this is sort of indicated here, but partially. And then you get a new species that then actually looks different than this one here, because it's maintained the duplication event. Now, when we look at evolutionary relation, when we look at homology-based inference, ultimately what we are talking about is what people refer to as orthologs, and the words are not really relevant. The moment that I have the same protein in 2 different species, I assume that they have the same function in these 2 different species. The moment I have a duplication here, I no longer know about this function. Possibly this protein is still sort of similar to this one because it sort of comes from there, but it is no longer constrained to have the same function, because essentially whatever you need this function for is already done by these 3. Right? You don't need the duplicated version. So it somehow is free to evolve. And this picture is very simple and very easy. The problem is, if you now imagine that sort of things would jump into an organism. So if you had jumping genes, right? Do you have jumping genes? So the way I asked that, you all by now know me long enough, right? Sure, you have jumping genes. First discovered by McClintock in corn and maize, where in fact part of the pattern of the different colors in maize come from jumping genes. So here we have an example of a bacterium or sea slug that in fact sort of gobbles up entire chloroplasts. So these are entire compartments from other species via a process called kleptoplasty. So some of you will know that klepto is a Latin word for stealing, or Greek word for stealing. So it's the craft of stealing. And essentially these entire in this particular case, chloroplast taking up this entire compartment keeps functioning in this originally intended way. So it gobbles up this compartment, it gobbles up all the genes with it, and all the functionality with it, too, in this particular case. So that's an extreme example. And now we know that, in fact, a lot of what is in the human genome is sort of jumping genes. What is the functional sense of that? Currently, we know as much as saying this is somehow for regulation. It's not turned into proteins, so it is not really turned into something that you see, but a lot is, and they refer to this as transposable elements or transposons. A lot in the human genome is sort of repeats that jumped out from various events in the past. Now, let's just make this story of homology-based inference slightly more complicated. So far, what I did is I looked at A and A2 prime and B and to B2 prime. What if I had another pair? And that other pair now would be in the same organism. Okay? So assume that I have a pair A prime, B prime, A2 prime, B2 prime at the same sequence similarity distance as the 2. So 1 prime and 2 prime have the same similarity to A, right? Which one would help you to infer function protein-protein interaction better? So if you knew this one, could you infer this better than if you knew this one, or the other way around? Or is it the same? The sequence distance, so this theta story here, is the same for the case. What do you believe? the story wouldn't be fun. I wouldn't tell the story if that would be the story. So it's just the other way around. So I show you again warm versus not warm. This is what people expect to be more conserved. And then I show you warm versus warm in blue. So the error bars are very high, but they do not even overlap the standard errors. You clearly see that, okay, there are fewer data points, everything is a little bit more complicated here, but what clearly is, at least for warm here, the ones that you assumed would be below are clearly above, right? And not only at one threshold of sequence similarity, throughout all the values that we can see. So we couldn't test at this point more similar ones simply because we did not have enough data, okay? Now, you could ask, this is something that is only warm. Here's an example for fly. You do see that the fly curves and the warm curves look different slightly, but you also see that ultimately, again, the blue ones here inside of the organism are always above the others. Why is that the case? We actually absolutely do not know. This is counterintuitive, absolutely the opposite of what one would expect. Maybe it sort of tells the story that when we duplicate a gene, we no longer really need it, but it actually helps to be double stitched. Maybe this is what we see. So having two versions of this that do the function, and having a version that does it slightly different, and in that sense can evolve a function that is more organism specific. So it still binds, but it sort of moves the binding site a little bit away. So that if I sort of replace that by a protein from a different organism, I could not easily eat up or take up this organism from a different species. It is true that the ability to sort of gobble up genomic material is different between different species. So bacteria can easily gobble up things. The worm that I'm talking about here is an extreme example. You essentially can inject, this is why the worm is such a great host for studying biology. You can inject any cancer, anything into that worm. The worm will express that. Any kind of DNA you inject into this worm, the worm will just treat that as own. We will not do that. So we will reject most foreign DNA. Anyway, so that is clearly an issue, and this means that homology-based inference will only get us so far, and we clearly need some prediction de novo. Can we predict PPI from sequence alone? We can, you know that. Here is, initially my idea simply was to predict two proteins interacting with each other. And the way I thought about that was to use a neural network that sort of looks at a region in one protein, region in another protein, and the part where they interact, and then essentially train a neural network. And so the first time I did this was by far the best failure I've ever produced in my life. This so didn't work. And the problem why it didn't work ultimately is because there's an ocean of false positives. So when two proteins interact, they have to be next to each other in space. So maybe if in a tube we would put particular proteins together, maybe they would interact. And in a cell they don't because they're not co-expressed, meaning they're not expressed at the same moment. Or maybe they're not in the same compartment. Or maybe there are other regulators that involve that. And this ultimately is the reason why we have to work on trying to reduce false positives. False positives, proteins that could not really meet and therefore couldn't interact. One way of doing that, for instance, is you predict surface residues. And if you do that and you say that proteins can only interact on the surface, then immediately you throw away three-fourths of all the false positives, right? Because the ones that interact is half times half is 1/4. It's a square effect, right? So half on one protein, half on the other protein leaves 1/4 of the residues that possibly are exposed on both and therefore could interact, right? That means essentially you quarter the false positives by this simple step. Yes, you're going to throw out some things that are wrong, because you make mistakes in your prediction of what is surface, but most of what you throw away is correct to throw away. Now, I'm not going to go into that. Then the next one would be to predict which residues are involved in these interactions. And this is what I'm going to talk about today. Jajun talked about it a little bit on last Tuesday. No, Thursday. I'm confused when. I'm even confused. Last Thursday, what was that? Yeah. Jejun talked about this one method. I'm going to talk about another method here. But this idea simply to have a method that does it, you immediately see that would reduce the false positives even further. Predict subcellular localization, again, for us was an important way to predict subcellular localization, just to do that, because about, let's say, a third of the proteins are nuclear. A little bit less than that, but then you reduce by a factor of 9 the false positives in the same sense, because you only need to pair proteins that are between different compartments. And then, you know, you try to predict proteins at substrate interfaces, you try to do better on domains, and then at some point you can put it all together, and that I'm going to talk about in the first lecture next year. Tobias Hump did that. Now first about the data collection. Again, binding are these two, but binding, in fact, in order to predict that two residues interact, I also need to define what is an interaction. That means some distance between atoms. So they must be closer than something. And we chose at this point here 6.5 angstrom. And then we get something such as this one here. We get amino acids. This is the less than expected, more than expected, in 3 different classes. So we are asking the residues at the interfaces of protein interactions. Do they differ between two homodimers, so two parts of a protein that when the same protein binds itself, internally in green here, meaning inside this protein, and externally, so that's the regular protein. So these in red is 2 different proteins that come together for some amount of time. Blue is slightly different. It's also it's not 2 different proteins, the 2 similar proteins, or the same proteins, and they are somehow closer together in the way they are expressed. So they don't really have to meet somehow, right? Now, what you clearly see is they differ. The big question is, so they differ. One way of saying they differ is that there is not a single one of these here where, in fact, all 3 have exactly the same value. You also see that all values here, all 20 amino acids in the 3 different classes, differ from 0. The background essentially is any residue in the same data set. So that means essentially the ones that bind look very different from the residues that don't bind. Okay, and that is true for all 3 of those. So the green ones, the internal binding sites, clearly differ from background, from random. I mean random as in you randomly pick protein residue on that particular protein in your data set. And you see that again blue differs from green, red differs from blue and green, so they all differ from each other. The real question is, the differences, are they significant? Are they meaningful? And typically, in order to sort of address this issue of meaningfulness, you employ statistical significance. Statistical significance, essentially you compute a standard error here and then see how much the distributions differ. The one way of doing that, one statistical process by which you do that, is the chi-square test. The chi-square test has some known problems. For instance, small data sets. We don't have small data sets. So this is the chi-square is very typically used in medicine, where you have data from 3 or 4 patients, and that then is complicated, because then it produces artifacts or produces results that are not completely reliable. So we have millions of data points. That really is not our problem. But another not so well known fact about chi-square is when the data set is too large, tiny differences matter. Sort of random differences, because if you sort of randomly choose a very large data set, it will be a Gaussian. And any deviation from the Gaussian is absolutely significant, right? This is somehow one of the problems of chi-square. Now, in our case, for those 3 classes, we see 10 minus 300. So this is way beyond significant. And the reason is this was sort of the limit of the computer. So it was just the end of its computational precision, right? So typically, 10 minus 3, 10 minus 4, minus 5 is when people publish. So 10 minus 300 is absolutely safe. Now, the person who did work on that, Yanayo Fran, was more skeptical he grew up in an environment where you have to be very skeptical, so he cross-checked his data again. What would you do in order to sort of... Yanai had the courage to argue, let's just do something very stupid. Let's just look at proteins that have a letter A or B in their name. Or let's just look at in one set I put all the proteins that were submitted by one author to some journal, and in the other one all that were submitted by two authors, and so forth. And this is totally nonsense, right? If you would still see that there's a significant difference, then something is strange, unless there is a code in the name, but there is no code in the name. Or unless there really is something between whether one author does something or two authors. And actually, there is a little bit in that signal, but that's a different story. But then he got to this issue of random subsets, and whatever he did, it was 10 minus 300. So now you come to the point where you essentially know that the statistician says, well, this is the right data set and it doesn't work. Why doesn't it work? Could it be because the data set is too large? Maybe. But ultimately, one argument in the chi-square test, you can apply chi-square only if the data sets or the sets that you test are statistically independent. Okay? We are testing internal residues and protein-protein interaction residues. They are not statistically independent. Proteins evolved. There's evolutionary signal that all proteins are connected. Absolutely not independent. So if that would be the problem, that would mean the end of statistics for computational biology. So is that the reason? We actually do not know. So I'm leaving you with a story, with a puzzle, for which we never found a solution. So strictly speaking, yes, we should not have applied chi-square, but there's no other test we could have applied to find out our result. And what it means, I do not know. We devised a different test, and that is sort of something that is very similar to what people do, very, very trivial. We take these three data sets, we take out some subset of all the samples in that data set. In some sense, we sort of repeat what you suggested, Lena, but we make a test out of it. So say we took 1,000 here from each of these classes. And then we take another 1,000 from the first. And we ask, by some Shannon entropy, Jensen-Shannon entropy, what is the set here most similar to? This, or that? If it is most similar to this, it is most similar to itself, then there is ultimately a proof that at least for a larger set, you can distinguish without machine learning, right? Just by simply this Jensen-Shannon entropy. So I'm going to repeat this experiment 1,000 times. And now actually I'm looking at 6 interfaces. So the green is still as before. The blue is as before, and the red is as before. And I sort of include some other classes, and I don't want to get into the detail. Ultimately, you see that the diagonal is clearly much higher. So all of these things differ. And now if you did the name or the length or the random, this would not be the case at all. Now if you did the trick with the name that I said, so you use all the ones with an A or B, then essentially they are equally filled, all of these. The numbers are the same all over. So clearly we can distinguish between these interfaces, and if we can distinguish, we should be able to sort of predict. And predicting means we have to get into a data set. So remember, some of you may remember from the whiteboard, the reason why we initially asked the question, do internal residues and protein-protein interactions differ, is because if they wouldn't differ, I could simply train to distinguish binding residues from non-binding residues. If they differ, I really have to explicitly train of protein-protein interaction residues. And so we have to build a data set of that. Those are the backgrounds. So we say 6.5 angstrom, larger than that. We're not going to go into this insert, I'm sorry. And we can do machine learning. How do we choose the input features? So if you, in a modern universe where you have a lot of data, record that or not. So the one-hot encoding, it would simply take the following idea. So you have 20 amino acids here, and one of these 20 has a value of 1. Every one all the others are 0. It's not 20, it's 21 because there's a spacer unit. And I have this running window, in this particular case 13 consecutive residues, and I want to predict whether the center residue, in this particular case here the P, is a protein-protein interface, yes or no. In brackets what you see here is P meaning interface, N means not. So that's why I have 2 output units. I could do it with 1. There is some reason why we use 2. And then essentially we decide which of these 2 units is larger. So if this one is larger, then the prediction is P. If this one is larger, the prediction is N. Here I have 2 examples for a prediction of P, 0.9 versus 0.1, 0.6 versus 0.4. In both cases this is higher, so in both cases P is predicted, but this is weaker than this. The signal is simply stronger. So yes, this would be the brute force way of doing it. What else could I do? So what else could I put as input?
Windows of fixed size.
That is a window of fixed size.
Yeah.
So I want to put in something on top of, so this is the step 1 or step 0, yes?
The length of a protein?
Yeah, that would be one that may be relevant because, well, let's look at the most extreme one. Very short proteins will less likely bind many proteins. So the number of binding sites will be more limited for a very short protein. This is one thing, yes?
Maybe some residue information like hydrophobicity?
Yes, biophysical features. We clearly assume that they will be relevant, and the biologists could sort of tell you maybe they are relevant, but you have to find your own way of coding it. But somehow you can try them. What else could you put in?
The number of the residue.
That is a very important point. So where in your protein are you? Now that point is important, and this is that requires some additional knowledge, biology, to sort of understand that point, because essentially when you assume that the protein ends here, so most proteins in fact have their ends somehow inside. Okay, this is one reason why where you are is relevant, because in a short protein you already see that there's not much you can do. If you want to interact, you have to somehow interact here, right? And here you cannot. So this already gives you some immediate way to sort of cut out some false positives. What else could you put in? Well, for those of you who were around in the lecture round 1, structure prediction, you essentially can put in whatever we predict as structural features. Secondary structure, solvent accessibility, the flexibility of a residue, B values. So all of this is in fact put into this system here. And the most important one, sort of the elephant in the room, is the evolutionary information from alignment. So rather than taking only one sequence, we do an alignment of a family and put in, rather than the hot encoding, we put in the conservation of that particular residue in that family. Because we assume that in the family, they all have a similar binding profile. So that if there is some biophysically relevant kind of letter that I need, I will observe the same letter throughout that family. And if not, then maybe either the biophysics is not relevant, or in fact that is not a binding residue. This type of binding, it has the biophysical features of a binding residue, but it happens to be not one, and that is what you see in the evolutionary signal. In fact, sort of all of these, disorder is another point that it was important. Some predicted aspects of function are put in. And then we sort of see how well we do. And the answer to that, what I show here is recall versus precision. And this essentially, sorry for the quality here, this is the curve without evolutionary information. Putting in evolutionary information, so by the way, this is random background. One additional point that I want to make, so this is the same curve as before, is just that I cut off here. This is only 3%. The last one was 100%, so it's way over there. This is why this looks so bitter. It is better than random. There is an issue that I'm going to skip over too. Then you put in the evolutionary information. This is what you had before, and this is what you get with evolutionary information. So you're doing way better here, and you're doing way better than throughout. Now, that brings up this issue of hotspots. You have a question?
Because you leave the disorder out in choosing the features. What do you mean by disorder? Because I'm kind of interested in, is it a disorder in structure? Is it in,
yes. So
I think this is a very important.
For the course of this lecture, I've always said that proteins fall into a unique three-dimensional structure, and that the detail of this three-dimensional structure is relevant for its function. That has been sort of my mantra, if you want. And it's sort of largely true. But then there's disorder. And disorder is essentially saying this whole thing is nonsense. There's a whole class of proteins that essentially appear to have the function of not forming a unique three-dimensional structure. What I mean by not unique means essentially they form something in some point, but they wobble around. At the next time point, they will actually look differently. And that is not against their function, that is part of the way they function. In some cases, let's call it orderly 3D structures induced upon binding. So they're sort of wobbly until they bind something, and then they look like a helix or like some regular structure. So the binding induces a regular 3D structure. And then, so if you think about it as inducing light, if you could shine light on it and observe it, you cannot, as I said. You have to make the crystals out of it. But if you could, then the disordered one, you would shine light at two different time points, so you would see different things. The moment they bind, two different time points, you see the same thing. So they adopt order upon binding. But natively, they are unstructured, disordered. And these, in fact, now you can imagine that one reason why this actually is helpful is because if you're more wobbly, maybe you can bind to two different surfaces. Maybe being more wobbly is a way with the same protein to bind two different substrates. And that is exactly what is happening. So some of these disordered proteins are absolutely important for binding different things. That is why this is also an important input. So there is this issue that you may bring up. How come that there are some residues here for which you predict very well? Very few. So the recall is very low here. But the precision is almost 90%. How come? Right? And ultimately that brings up this issue of hotspots. Hotspots, PPI hotspots, the idea is essentially that you have a particular residue, so you have an interface between two proteins, protein A and protein B, and you see there's a lot of interacting residues, but a hotspot, and they do not have to be close in sequence or 3D, these could, if these would be hotspots, that would mean that they contribute to this interaction energy much more than the others. So in the way I draw that here, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 residues that sort of maintain this interaction. And they would be hotspots if their contribution would be much more than 2 by the 10, or 1 by over 5, right? So it would be much more proportional. So if all would be equal, then each would contribute 1/10 to the energy of binding, right? And if there would be some, say, that contribute, say this one here contributes 10%. Oh no, that's a bad example. That is 1 over 10. So if this contributed 40%, and this contributed 30%, so these two together would contribute 70%, then they would be completely outliers, right? And then we would possibly call them hotspots. So why would they be relevant? Yeah? Different question.
I'm sorry, but how do you quantify?
That's exactly the issue. Thank you very much, Stanislav, because ultimately this is, your question is the entire crux of the story. So here I would label 1 to 10, and here I would show the contribution to the energy, delta G of energy of binding, for instance. Okay, and then you sort of see a curve that looks exponential. And if you see an exponential, you may argue, well, one thing is absolutely clear, they're not contributing equally. Some contribute much more than others. And depending on how sharp this exponential is, you would say they contribute much more or something like that. Now, when can you ask, at what point, what must be the difference here in order, the delta and the delta G, at what difference would you say they contribute much more, right? And this is essentially why has been a big discussion in the group over this topic. Are there hotspots? So some people argue this is nonsense. Yes, it's exponential, but wherever you put the threshold is meaningless. Some are more than others, true, but none are really different. There's no biophysics. There is a gradual decline. It's just exponential, but it's still gradual, right? And you cannot define, so what you're asking me for is the definition, you cannot do it in a reasonable way. Let's call them the hotspot agnostics. And then there are the sort of hotspot believers who say, well, you know, if this is a factor of 4 or 5 or 6 or something like that, if 2 in 10 contribute 70%, then this is a very, very, very strong effect, and I could argue they are different. Now, this question is not only academical, because all the drugs will target exactly hotspots. Because if you want to sort of knock out something that is a disease interaction, and you know that these two here contribute most to that disease, you will try to knock out these two first. That's the most effective way of attacking, right? You attack first the ones that are the most energetically important ones, right? So, yeah?
So the hotspots are certain, are not random in every protein?
So are there hotspots in every single interactions? Well, that again, so the believers of hotspots would say yes. The agnostics would say, because in fact you have different exponentials with very different shapes, so it's not always a factor of 5. Okay, in that sense. But every interaction has some, so every interaction essentially is, every interaction I've ever seen is sort of characterized by an exponential.
And it has the same residues that are hotspots.
Oh, so
So every interaction.
now you're opening a can that is actually not. Let me give you the generic answer. So for the interaction between A and B, yes, it is always the same hotspot. That's why the drug targets exactly that residue. And that answer is true enough because this is exactly what people who develop drugs do. And all the drugs you have ever taken had some success. And ultimately, because they essentially always are. Yes, Dan?
And about the energies, do biologists measure them directly, or do we infer them from something?
Well, I'm not entirely sure what you mean by. So you cannot go there with a little thing and measure the energy. All right. So there's actually this is also not quite true. So actin is a muscle fiber, and in the muscle fiber that pulls, they actually hang really manage now to have micro weights and can really measure it. So in some cases you can really measure proteins like in that sense. That is not what they do here. But it's essentially, you measure pretty directly the effect of the energy. Let's put it like that. There's not much of an interpretation. So there's very little doubt. I mean, give or take some factor. But you know, in this curve, a factor 2 is totally meaningless because it's sort of the same factor of 2 throughout, right? The relevant thing here is the curve. Yes?
And are the hotspots, like, can we infer hotspots based on homology? Like,
so now we get into the point. So the agnostics say there are no hotspots, then the answer to your question is absolutely no. For the believers, the answer is yes.
not hotspots, can we infer energy levels based on homology, let's say?
Yes, but in fact now again, so the problem is that so everything is fine as long, absolutely yes, as long as you have these two as they look. The moment you move something a little bit further here, or you put some other positive charge here, this may influence that, and then things get a little bit more complicated. Because something that happens here can put some strain onto the surface, and that slightly changes the distances. And then that could alter, right? And it could alter in a way in which is not true that they all are just because it's sort of pulled away a little bit, that all interact less strongly. It could be that depending on where the force comes from, you could imagine that you pull here more, then these would be more affected. You pull here more, these would be more affected. So the conservation, the homology-based inference of this energy would differ between here and there, depending on what else happens in the molecule. But largely, yes. Okay? Now, one way of thinking about it is maybe there are some that we can predict very accurately because these are the hotspots. Maybe we have never trained that, because all we trained is essentially proteins, all the protein residues, nothing else. We have never labeled them as hotspots or not. But maybe the machine learning actually picked out hotspots. And if that were true, then all I need is some experimental data set of hotspots. And I got that from a person who sat next to him. And in the same image that I have been showing you from the GP120 and CD4, that experimental group sort of changed many of these residues and found out which ones are the most important for that interaction, and they are shown in red. And this is what Yanai predicted. So never trained. He's just looked in the curve that I showed you before, simply at the ones that are most strongly predicted. That's all. He has some threshold that he chose before seeing this data. Ultimately, when you see this curve, you sort of cut off here, right? Because this is where it drops until here sort of looks okay. You could cut off here, but I believe he cut off somewhere here. And that is the purple. Here you see them on the structure. We didn't have the structure to do this, but you do see that in some cases here, for instance, you miss one. In some cases you overpredict. Where's one overpredicted? I don't see that at the moment. Yeah, exactly. Here, that is overpredicted. I made a mistake. That is overpredicted and that is missed. Thank you. Exactly, but it's remarkable. That was the first case he ever tried. And in fact, in this particular case, he tried that in the last minute for his thesis, because when Hendriksen had those data in his pocket and was on his thesis committee, and he just wanted to sort of derail him. Didn't work. In this particular case, the method worked very well. And then he sort of gathered some data, and for some data, these numbers look different, but for some data set that he could get his hands on, overall, the method could predict hotspots without ever having a concept of hotspots. And the argument simply was, if we can predict hotspots, they must differ. So there must be some feature in them. Maybe it's just the strength. But the strength creates a reality that leads to a consistency in the machine learning that allows the machine learning to predict something without having learned this concept, right? And for us, this sort of was a proof. Now, let's briefly get into this issue of hubs. So hubs are proteins, PPI hubs are proteins that interact with many other proteins. So since essentially we have a method that predicts which sites on your protein or which residues bind, you could suspect that you can predict hubs. How would you do that? So date and party hubs, introduced by Mark Vidal. Date hubs use one binding site to bind to many proteins. Party hubs use many binding sites to bind to many proteins. So we would somehow expect something different. Now, here is what we see. We're looking at non-hubs in the front and date hubs. And essentially, this is the number of binding sites you sort of see. And you do see that the party hubs look very different from non-hubs and date hubs. Yes?
Can you train a classifier on the for the regions that can interact not with only one, for example, protein?
Yes.
For example, build a regression model.
So essentially predict.
Yeah,
yes.
Predict a number of...
Yes, so Yanai has, this was submitted, it was never, he submitted this many times, it was never published, but Yanai later published this, in which he did exactly that. So he coupled this regression with experimental data to show, in fact, that he can predict, and then he experimentally followed up on those predictions, and it turned out right, and that he published not that long ago. Yes, you can. This is just the same issue. Now, by the way, there is a flip side to that story, and the flip side is essentially the disordered story. There's a particular type of disorder, and I don't want to get into that, but this type of disorder is sort of indicative of date hubs. So now you essentially have two mechanisms. One, you use disorder to predict date hubs. Another one, you use binding sites to predict party hubs. So this is essentially, you now go a step further, the regression model that later led to the regression model.